Hello, and welcome to the notes on cell transport for Nat and Phys. We are going to move into part one of those notes right now. And in part one, we'll talk about passive transport and diffusion and osmosis. At this point, hopefully, we've already done our student kinetic movement activity. So you have a little bit of background on that, and we'll just be reinforcing that and making sure that's in the notes here. So, kinetic energy and motion. So one thing we learned in that activity is that all molecules have some kinetic energy. And kinetic energy is just energy of motion, right? So all molecules, unless they're at absolute zero, which is the point at which all motion stops, so they don't have kinetic energy, they're either moving around or they're at least vibrating a little bit. And that's kinetic energy. And that we can interpret as heat. The more kinetic energy they have, the more they're vibrating, the more heat they have. Or the other way to think of it as movement. Either way works, but we have some movement of these molecules. Thus, they're constantly moving, but it's in a random direction. They don't like say, I've got energy, I want to go to the moon, so I'm going that way. No, they got energy, they're just running, bouncing around until they hit something, then they bounce off, hit something else, and keep bouncing around. Random directions, and just pure physics, what they're bouncing off of. Now, this has the effect of causing them to spread out, because if they're tied up, packed up in this corner like this, and they bounce into each other, they're going to bounce around and they're going to start to spread out around the screen, right, as they go. Just like we saw in the food coloring in the water and in our experiment with student motion, it spreads out, which essentially means they tend to move from areas where they're highly concentrated and they have a net movement to areas where they have a lower concentration. So from high to low concentrations. And that causes them to spread out until they're equally spread out and they reach what we call equilibrium. That's what happens to all molecules, regardless of whether they're in a living system or not. What I wanted to focus on more here is to make sure we have a really solid understanding of the motion of these molecules, because they play a huge role in how our cells and our tissues and our organs regulate the amounts of materials we have to make sure we have the right amount of calcium in our cells or outside of it, the right amount of sugar in our cells or outside of it, and all that kind of stuff. It's going to come up over and over again throughout the course of the year, so we want to make sure you are really well grounded in this. So it's a little bit of review of what we did in bio, take you a little bit deeper, and make sure you really have this down, because it's a fundamental process that happens in our body. All right, so let's start with passive membrane transport, or diffusion. Passive means no energy. Okay, if you're just sitting there and watching this, not taking notes or even thinking about it, you are doing passive listening right now, right? You're not doing anything, as opposed to active listening where you're writing notes, repeating back what I'm saying to yourself, thinking about it. That would be more active note taking. But if you're just sitting and listening, it's passive. You're not putting much energy in, you're just letting it flow. And that's what most movement in the human body is. It's passive. And there's two types of it, or a couple different types of it. First type we're going to talk about is simple diffusion, and that is where very small molecules that are non-polar or lipid soluble are able to pass right between the phospholipid membranes of cells. So the cells can't stop, the cells can't control it. Alcohol is an example, right? It can pass right between those cells, right through the phospholipid membranes. They're small enough that they can squeeze through those tight little gaps between all those phospholipids, and they are nonpolar, like the lipids are. Lipids are nonpolar. They don't have charges like water. Water is a polar molecule. It's a really small molecule, so it'd be small enough to pass through, but because it's got a charge, it can get repelled by those phospholipids. So if it's nonpolar, no charge, so it's lipid soluble, it can dissolve through a lipid, and it's small enough, it can go right through the lipid bilayer. Now, it's still going to follow that basic force of moving from high to low concentration, even though, so it's able to pass through the membrane, but it's still going to go from high to low concentrations. So think about oxygen, right? Your cells are constantly using up oxygen and converting into something else. So you're always going to have a lower concentration of oxygen in your cells because you're using it up, which means there's always going to be a higher concentration of it outside your cells, meaning oxygen is going to want to diffuse into your cell. And now let's switch over to the whiteboard and we'll give that a try. All right, so just like to talk a little bit about concentration gradients now, and I like to think of them kind of like a hill. If I draw a hill on here, all right, we got a high spot and a low spot, right? And if I were to draw a big old boulder on the top of that hill, okay, it's going to want to roll downhill, right? Gravity is going to take it. The rock doesn't need to expend any energy. It's just moving like nature intended. It's going to go on down the hill. And this is basically like diffusion. We go from an area of high concentration, and that's what the brackets mean, concentration, to an area of low concentration. Does not take any energy to do that whatsoever. Okay, at least any energy on the part of the cell, I should say. It does take energy. The energy that's inherent to that rock right there is going to move it, and it's going to tend to move from high to low. So anytime you're given a situation where you're given the concentrations of material inside and outside the cell, you can think of it, it's just going to roll downhill. It's going to go from wherever it's highest to wherever it's lowest if it can pass through the membrane. So you can kind of think it like, a little gate right here. If it's able to pass through that, then it's going to roll downhill. Okay, it can fit through that little gate, no problem. This would be like a very small molecule passing, small nonpolar molecule passing right through the phospholipid membrane, which we called simple diffusion. So that was simple diffusion. Things are able to move down the concentration gradient from high to low with no real impediment to their motion. They can go in or out of the cell, whichever direction the concentration gradient's letting them roll. And since the molecules are small and nonpolar, they can get right through the phospholipid membrane, no problem, no big deal. But sometimes you have things that can't fit through the phospholipid membrane. We got things that are bigger or are polar. They can be pretty small things, but are polar, like different ions. We need lots of ions moving across our cells at various times for muscle movement and nerve signaling and things of that nature. So when we have really big things or polar things that can't fit between the phospholipid membranes, we can still let diffusion power that motion. They're still going to want to roll downhill, but what we have to do is we have to provide them with carrier proteins or protein channels. So proteins that are just the right size and shape to let the molecules through, or they can open up to let the molecules through at certain times. So carriers are integral membrane proteins that show specificity for certain polar large molecules, including sugars and amino acids. So they're just the right shape to allow certain things through, but not other things. Okay, and so let's go back to the whiteboard and I'll show you some of those. So now we've got the same situation again. We've got a high concentration. Let's pretend it's outside the cell, and a low concentration inside the cell. And this rock wants to go in. That's great if there's no barriers, nothing there. But what if we put in a wall? It's too big to fit through the phospholipids, or it's polar, so it can't get through the phospholipids. It's not going to go anywhere, right? So simple diffusion won't work in this situation. The cell needs to give the molecule a little bit of help. And that's why we call it facilitated. Facilitated, can't spell, diffusion. The cell needs to help it out. So in a situation where the molecule is too big or too polar to fit through, polar and can't go through the phospholipids, the cell can provide help to it or facilitate it. One of the things it could do, it can provide a doorway or a tunnel. Let's call it a tunnel here, right? Looks like a rock. It's supposed to be a tunnel that rock can go through. So imagine that's just the right shape for that rock to go through, and that rock then can travel through the tunnel and on down the hill. So that's one thing that we could do. Okay, that is called facilitated diffusion. We just provide a tunnel that's just the right shape for that rock to get through. And that's one way that the cell membrane can control things. But what if we do a tunnel that's that shape? Is that rock going to be able to fit through there? No, it's going to run into it. It's going to bang up against that. It's not going to get through. So that is another way that we can do a facilitated diffusion. Those protein channels are just the right shape to let specific molecules through, just the right shape to let water through, just the right shape to let calcium ions through, for instance, or sodium ions through, and not other ones. Okay. Another thing we can do is we can do it like a door. Okay, I'm going to try my best to draw a panel door here. Okay, so we have that door, and it is big enough, let's say, to get this molecule through, but it's closed. Okay, we have something like this for glucose. We talked about glucose in the first unit when we talked about negative feedback loops and regulation. And when your blood sugar gets high, your pancreas sends out insulin, which tells your cells to suck in glucose. What it's actually doing is that insulin is kind of like a little key. That's a key that unlocks that door, and then whoop, what it will do, it will open that door, and then the glucose can come in. Sorry, I don't know how to animate an opening door. But then when the insulin is no longer being secreted, that door closes and you keep that sugar out of the cell. And the other sugar that's coming up behind it, all that extra sugar, stays out there when you get the amount of sugar in the cell that you need. And you got the blood sugar outside the cell at the right levels. So that is also facilitated diffusion. It doesn't take the cell any energy to provide that door. Okay. All it has to do is open and close it. And that's one of the nice properties about proteins. They can change their shape. And this particular one can recognize the hormone insulin so that it knows that will cause it to change shape and let more glucose through whenever it gets unlocked. And that is also another form of facilitated diffusion. So we have regular diffusion or simple diffusion, where the molecule is able to pass right through the cell membrane, nothing to impede it. It's small and it's lipid soluble. We have facilitated diffusion where there's a tunnel that's just the right size and shape to let certain things through. And we have another type of facilitated diffusion where we have a door that we can open, open or shut to allow things in down the concentration gradients. Again, the cell's not actually moving the molecules. It's just providing a gateway to get in and out. And just to kind of sum up everything we've talked about with passive transport or diffusion so far, we've got our plasma membrane made out of phospholipids, okay? And we've got a bunch of different proteins embedded in it. And I'll put the pointer on here. So we can see here we got lipid soluble, small lipid soluble things, and this is simple diffusion. Okay, we got more of them outside than in, so we go from high to low. And since they're really small and nonpolar, they can pass right between the phospholipids. Can't do anything about it, like alcohol. Okay. Other things, these are small molecules as well, but they're lipid insoluble, so they can't get through over here. But what we do is we provide a carrier protein or channel protein, and that's just the right size and shape to let those things through, and this would be open all the time. So diffusion could happen all day long, every day with a protein channel, okay, or channel-mediated facilitated diffusion. Facilitating is helping things move across the membrane. Over here, we have some that aren't open all the time, okay? That's carrier-mediated facilitated diffusion. So once again, we're providing help, we're providing a doorway, but this door we can either open or close. Over here, this is just open all the time. This one, we can open or close. So here we got a large solute, lots of it out here still. So it still wants to go from outside to in, down the concentration gradient from high to low. But here, it's going in here, and this carrier will let it through. And you notice once it lets it through, it changes shape, lets it through here, but doesn't let it come through on the other side. So this one has the ability to close or open, depending on what you want to do, but we're letting them through. Okay. So one more type of passive membrane transport I want to talk about in this section of the notes, and that is osmosis. It works basically the same as simple diffusion, okay, or facilitated diffusion, right? You're still moving things from high concentrations to low concentrations, so down the hill. However, in this case, it is water moving across the membrane from high to low concentrations. And so all the principles are the same. We got the molecules, they got to get across the membrane. They're going from high to low concentrations. There's a term in here called tonicity, which is how a solution affects cell volume. but it can get a little mixed up in folks' heads. So let's take a look at a picture that'll hopefully help us understand those differences. So here we got the first U tube, U as in the letter U. And you can see here we've got water on both sides in the blue, and then we got these little red molecules of solute, could be salt, sugar, anything, okay? And we have a membrane between them. Now, if this membrane allows both the solute and the water to pass, solute, the red dots, have a high concentration over here and a low over here, so they're going to want to go from high to low, and we see that with this arrow. Now, the water, the higher concentration of water is over here, right? More water and less solute, okay? Basically, this is closer to pure water. You can't get any more concentrated water than pure water, so water with nothing else in it. The more and more stuff you add to the water, the less and less concentrated the water is. The other stuff gets more concentrated, right? So you can think of this like slightly salty water, and this would be like ocean water, right? This is a higher concentration of water, less salt, a lower concentration of water because we have more salt in it. So the water wants to go from high concentration, where there's larger amounts of water compared to the solute, over to here. And in a situation where the membrane allows them both to pass, solute moves one way, water moves the other, and they reach equilibrium on both sides, no problem. Okay, that happens. But one interesting thing that can happen is if you have a membrane that allows water to pass through, but not the solute. So we're starting with the same situation. High and low solute concentrations, high on the right, low on the left, but the solute can't pass. So there's no way to balance out by moving the solute. The only thing that can move in this situation is the water. It's still able to pass through the membrane. So water still wants to go from high to low. So it moves from high to low until it balances out those concentrations. And you notice it creates, the concentrations are the same, though the levels aren't. That osmotic pressure is strong enough to resist the force of gravity in this situation to try to get close to equilibrium in the system. So that's osmotic pressure. This is what happens when kind of plants wilt, right? If you don't water them, they'll wilt. What's happening if they got plenty of water? That drives. It's more like this kind of situation. Plenty of water. The water gets driven into the cell membrane, and that cell membrane then pushes out against the cell wall of the plant cell, kind of like when you have enough air pressure in a tire. This osmotic pressure keeps those cells rigid so that the plant can stay upright. If you don't have enough water, the water concentration drops. There's not going to be as much osmotic pressure forcing water in. The pressure inside the cell drops a bit, and the plant will wilt. So you'll see grocery stores will use this trick when they're trying to sell you like fresh veggies, like celery sticks. They usually package them, if they want to do like little snack packs, they'll package them in a cup of water. Right? I want you to think about that for a minute. Why would packing them in a cup of water help them sell these celery sticks as being fresher longer? So let's see if you got that question right. The reason it works is because by packing in a cup of water, it's doing this situation, right? It's going to force, water's going to want to get into the cell because pure water outside, inside the cell, there's other stuff floating around, other solutes, so the water's going to flow in. Osmotic pressure, it's going to fill up that cell and make that cell nice and firm. So that celery is going to be crispy, not the kind of floppy celery if you keep in your fridge for a while. It's still green, it's still good, but it's kind of floppy because it's lost that water. So that's what's going on right there. And that will end part one of our notes on cell transport, dealing with passive transport. So we have simple diffusion is passive transport. That's molecules going from high to low, passing right through the phospholipid membrane, no real impediment. So they're small and lipid soluble. Then we've got facilitated diffusion, where the cell has to provide some sort of a gateway or doorway with proteins to let the materials in or out. But the cell still doesn't have to use any energy to do that. Still the energy of the molecules moving them uphill, downhill from high to low. You just got to open the door, so to speak, and let them walk in. And then the last one is osmosis, which is also passive transport. Same thing, except we're dealing with water specifically moving across the membrane. Okay, and water will generally move. If water and solute can both move, water will move in the opposite direction of the solute. All right. We'll see you next time when we talk about tonicity.